Students, welcome back. So, in the last video, we were talking about the different characteristics where Mendel studied in green peas, right? So, that is the garden pea plants. Okay, these are the 7 characters what we find. Of course, here it is not the seed coat color, it is the flower color. So, flower we get there is a violet flower as well as white flower. Okay, so this is not green and white, it is violet and the white flowers. Okay, next is we are going to discuss about the next. You can see here the pea plants, these are the tall plants, stem height, and there are plants, dwarf plants or short plants. Okay, next will be, suppose if you take the other trait, seed shape, you can see here they are the round shaped and here the wrinkled shape. That means they are not smooth. Okay, that is another seed shape. So another example, flower colors. So he found that two contrasting flower colors are there. That is the violet flowers and the white flowers. Okay. Next, there are certain terminologies we use in genetics and the basic terminologies we must be aware of. Right. So what Mendel called the factors, they are nothing but genes. Right. William Johnson is the one who coined the term gene. Right. Genes are nothing but, or whatever the Mendel used the term factors, nothing but they are the functional unit of heredity responsible for the expression of character in the progeny. So they are the one basic unit of heredity. Then locus. Locus means location. Say for example, a particular gene is present which is controlling the trait. It is present on the same location on the parental chromosome, that is the paternal chromosome, father chromosome, as well as on the mother chromosome. Say for example, height of the plant is not only present on the pollen grains in this particular position, it is also present on the same position. It is not like the gene is here and other counterpart allele is there. So these two forms of the genes we call them as alleles. Okay, allele is the alternative form of a gene for a contrasting character. Say for example, simply I am taking the example, this one is controlling the tallness in the plant, this one is controlling the dwarfness in the plant. So that means they are slightly changed genes, but they occupy the same location. So contrasting characters are controlled by alleles present on identical locus of the homologous chromosomes. Homologous chromosomes means, say for example, in case of human beings, we are having the 46 chromosomes, and 46 chromosomes are being classified as a 23 pairs. So that means the chromosome number 1 of the father and the chromosome 1 of the mother will be of the same length, and as well as they contain the same genes on them. Right. So that is the simplest explanation, and later on you are going to study about this homologous and then the sister chromatids, non-sister chromatids and all. Then comes phenotype. So it is the external appearance of an organism due to the influence of genes. So genes are controlling the phenotype. So visible trait is considered to be the phenotype, and the environmental factors also affect the phenotype of an organism. Then the other term we use is genotype. It is the genetic constitution of an individual responsible for the phenotype. That is what is called as genotype. Then phenotype ratio, it is the proportion, correct proportion of the phenotype in a population. Okay, so how many tall plants are there, how many dwarf plants are there, so in a total number of plants will be called as phenotypic ratio. Then genotype ratio, so what is the genotype ratio? Whether what we say, the same homologous genes are there, or what we say homozygous genes, not homologous, sorry, it is homozygous genes are there, heterozygous genes are there, whether homozygous dominant are there or homozygous recessive are there. So that proportion is called as genotype ratio. As we work out the Mendel cross, we are going to find out. Then homozygous is the individual having the identical genes in an allelic pair of a character. Example, say Mendel used the alphabets to represent the genes. It is not possible to remember the sequence of nucleotides for a gene on the DNA. It may contain several nucleotides, maybe 1,000, 10,000, 15,000, right? So one gene may contain many, and it is not humanly possible for us to remember in the sequential order. So that's why what Mendel gave, it is only alphabets, and alphabets also he gave in a scientific manner, in which capital alphabet will be used for a dominant trait, and the small letter or the lower case is used for the purpose of representing the recessive trait, right? So homozygous condition means both will be of similar alleles, that is capital T or capital T and small t. So that is what is called as homozygous condition. Then the individuals having unidentical genes, so slightly changed genes in an allelic pair for a character, is called as the heterozygous condition. That is capital T and small t. Here we cannot find T on the DNA. We find only the nucleotides, but only for our convenience we represent it in the form of alphabet. Just like we represent, instead of calling your name during the attendance, we assign you the roll number and simply we call 1, 2, 3, 4 like that. Here also the Mendel used this technique. Now this is how, for example, father is having, say this is the what we say human genotype. Okay, human genotype, you can see here the first pair is having both lengthier, then a little bit shorter, shorter, keep on reducing up to 22, which are controlling the body characters, or these are considered to be autosomes. And of course the last pair, which is what we say the allosomes or sex chromosomes. Of course in case of male the pair is not identical, or there is a very short Y chromosome compared to the X chromosome. So the female is also having the 22 pairs, and the last pair is a similar pair, identical pair, where both are XX chromosome. Okay, so these are the chromosomes. The last pair, the 23rd pair, is going to determine the sex of the baby, that we are going to discuss a bit later. Now, when the meiosis, when during the reproduction, when meiosis takes place, out of this pair, only one of the allele will be transferred to the gamete. Say for example, the sperm, here in case of father, and the egg in case of mother. So not both will be transferred, only one will be transferred. Okay, so that means out of 46 chromosomes, only 23 chromosomes are there in the sperm, and 23 chromosomes will be there in the egg. Right. So in a pair, only one chromosome will be transferred or inherit to the gametes, and when the gametes fertilizes, the original number is restored. Right. 23 from the male gamete, 23 from the female gamete, and it will become 43. You can see the color differentiation here also. This blue color father, then this pink color mother. You can see that pair also, they are the similar chromosomes, right? So that's what we need to be very careful. Then when the Mendel crossed the pea plants, first he has started with simple experiments. Even though there are plenty of characters are there, but his observation is focused on only one particular character, say the height of the plant. Okay, so height of the plant, he found that two contrasting traits. Out of that, one will be tall and the other one will be dwarf plants or shorter plants, right? So if the inheritance of one gene, because these traits are controlled by genes, inheritance of one gene is studied, such type of cross we call it as monohybrid cross. Okay, so Mendel selected the plants having a pair of characters, that is tall pea plants and short pea plants, dwarf pea plants, and he selected pure plants. Pure plants means many generation he self-pollinated and observed that the trait is not differentiated, or observed that the trait is not deviated. So it is simply moving from one generation to another generation. Say for example, a tall plant is crossed with a tall plant, you get tall plant. Once again he crossed with a tall plant. Once again the progeny will be tall plant. And then the short plant, and short plant is crossed, he got only short plant. He crossed it with short plant. Once again he got short plant or dwarf plants. So next also he crossed. So many generation, maybe 5-6 generation, if the alleles are not segregated, alleles are not going to be changed, or the trait is not going to be changed, such plants are called as pure breeding plants. Pure breeding plants or true breeding plants, right? Once these true breeding plants, that means they are homozygous in condition. Homozygous in condition, they are crossed. He got the result like this. Okay, a dwarf pea plant is crossed with a tall pea plant. In the F1 generation, for his surprise, he got all the tall pea plants. Right, only one parental variety is observed. Okay, when he selfed among themselves, among the F1 progenies, he got both the tall plant as well as the dwarf plant. So both are astonishing thing. First, when he crossed this short plant with a tall plant, none of the plants were intermediate or none of the plants were short plants. All are only one parental type, that is tall plants. Okay, somehow that was inherited. And the next, when he crossed the tall plant and tall plant, to his surprise, he got tall plants as well as the dwarf plant. Okay, so that means what he concluded from this is the allele responsible for dwarf plant is also inherited to the tall plant, but it could not be expressed because the allele responsible for the tallness is dominant over the other allele, that is the short plant. That's why the phenotype is expressed as tall plant. And when the allele responsible for the dwarfness is just combined in the homozygous condition, he could get the shorter plant or dwarf plant. Okay, so there we come across dominant and recessive trait. And he did the same experiment for other characters. So here he has taken the flower color. Violet flower is crossed with white flower. Once again he got only one parental variety, that is violet color. So that means this is a dominant trait. Okay, so these are the true breeding plants, and when the parents are crossed, he got only one parental variety, not the other one. So this is a dominant trait. But once again, the F1 is self. A violet plant of the F1 is crossed with the violet plant of violet flower of the F1 generation. He got what we say 3 violet flowers and 1 white flowers. So both the parental varieties are present in this. So here in the third one also he got the round seeds crossed with wrinkled seeds. In the F1 generation, only one parental variety, that is round color, is formed, and in this second he got 3 round and 1 wrinkled in the F2 generation. You can see in all these three things in a monohybrid cross. Monohybrid cross means the inheritance of single gene. Okay, in F2 generation, out of the 4 plants, 3 were tall plants and 1 dwarf, that is 3:1. Then here 3 violet flowers and 1 white flowers. Once again 3:1. Here 3 round seeds and 1 wrinkled seeds, that is 75% will be of dominant trait and 25% will be of the recessive trait. So this ratio which is being seen, it is a phenotype. What we are seeing is the 3:1. So 3:1 will be the phenotype ratio of F2 generation in monohybrid cross, right? So that is the phenotype ratio. Now the same thing clearly individually we are explaining. A tall plant crossed with dwarf plants, both of them are true breeding or both of them are pure breeding plants, that means homozygous in condition. So he got in F1 generation both tall plants, they were just self-pollinated, right? And he got 3 tall plants and 1 dwarf plant. So the phenotype ratio of F2 generation in monohybrid cross will be 3:1. Okay, next this is how it could be obtained. So what is the reason, basis of that? Because he is a mathematician, he worked out by using these the alphabets for that particular trait, right? Say for example, a tall plant, we already know that which is receiving both the alleles, that is for one from the father, that is pollen grains, and one from the egg, right? So that means it is in a pure condition, that is homozygous condition, capital T, and the dwarfness is being represented by small t and small t, right? When gametogenesis takes place, when gametes forms, there is a meiosis. Meiosis will reduce the chromosome numbers to half. That means the alleles are also going to be reduced to half. Out of this pair, only one allele will be transferred to the pollen grain, right? Say for example, if this is a male plant, then so only one T is received by the gamete. Here one small t is received. When these two combines, fertilizers, they will restore the diploid condition, or both the chromosomes are present here. So one will carry the capital T and one will carry the small t. And since the capital T, the allele is a normal allele which is dominant over this altered allele, that is this small t, we find that in F1 generation all the plants are tall plants, right? So when this mating among these F1 generation takes place, here we find that there are two different alleles. So that means 50% of the pollen grains or the gametes will receive one allele, and the other 50% will receive the other allele. Here also 50% of the eggs will receive capital T, and the 50% of the allele will receive small t. Okay, so there are four types of gametes, or two types of gametes from each parent is produced. Okay, now if this T combines with this T, we get capital T, and if this T combines with this T, we get capital T, small t, and if this T combines with this T, we get capital T, small t, and if this T and this T combines, then we get small t, So like that four plants are produced, and already we know, or what Mendel observed is capital T, that means the normal allele is dominant over an altered allele. So that's why this allele, if it is present, it is going to express its phenotype. So thereby here capital T, so this is homozygous tall. And these two plants, capital T, small t, these two plants are also tall because the capital T is dominant over small t. That means these two are also tall, but they are in heterozygous condition. Whereas the small t and small t both are in recessive condition, where an altered allele will produce less enzyme responsible for the growth, and thereby the growth is reduced and will become a dwarf plant. So by this way, the phenotype ratio will be tall is to dwarf will be 3:1. What is the genotype ratio? There are three types of genes are present. One is homozygous dominant alleles, then homozygous recessive allele, and heterozygous dominant alleles. So that means capital T, small t, and small t, So that will be in the ratio of 1:2:1. Understood? Okay. So the same thing in a simple way. Tall plant, dwarf plant. Tall plant will be having homozygous dominant, that is capital T. This is homozygous recessive, small t. In F1 generation, one of the alleles. So all gametes will be produced by this will be capital T. All gametes will be produced by this will be small t. In the last slide you have seen, right? So then it will become capital T and small t, but all are tall, because tall is dominant to the dwarf. Then this will produce heterozygous two types of gametes, right? So capital T and small t. Here female is also produces capital T and small t. These two, when they combine, capital T, V, T, that will produce tall plants. Capital T, small t will also produce the tall plant, and the capital T and small t, that also produce the tall plant, whereas small t will produce the dwarf plant. So that is the phenotypic ratio of F2 generation in monohybrid cross will be 3:1, whereas the genotype ratio is 1:2:1. So here 1 refers to the homozygous tall, 2 refers to the heterozygous tall, that is capital T and small t, and this 1 refers to the small t and small t. Then the same monohybrid cross is being carried out for other traits also, where we have taken one more example. suppose the color of the seeds, yellow is dominant over green, and as usual by convention yellow color is being referred or is being assigned capital Y, and the green color is referred with small y, and never write a single allele, because diploid organisms there will be two copies of the allele will be present on two chromosomes, that is the one is on maternal chromosome and another one is on paternal chromosome. So diploid organisms always carry two alleles for a particular trait. Don't write yellow means single y and green means small single y, right? You have to always write two alleles, right? So suppose these are the two alleles parents. When the gametes are produced, only half of that allele will be passed, that is only single y and single small y, and in F1 generation when the fusion takes place, capital Y and small y, right? Then this capital Y is going to express, that is dominant over small y. Then in the F1 selfing, when the capital Y and small y, the round seeds are crossed, but their genotype is different, they will produce two types of gametes, that is capital Y and small y, and another plant also will produce the same capital Y and small y. We find the same, that is capital YY, small yy, capital Y, small y, capital Y, small y, small y, small y. Okay, so it is just like a tall and what we say the dwarf plant, the same thing is there, right? So here we got the phenotype ratio 3:1, right? So 3 phenotypes and 1. Out of that 3, capital YY will be 1, capital Y small y will be 2, and small y will be 1. So the genotype ratio is 1:2:1. So it is another example. Okay, I think by this time you know. And another example is the purple flowers or violet flowers crossed. We are going to get the same ratio. Same ratio will be there in both the cases. Okay, so here also we get the same ratio. This is only the examples. Then after concluding the monohybrid cross, Mendel proposes certain theories of inheritance or laws of inheritance, right? So the first law is law of paired factors. Because Mendel does not know the genes, so he called them as factors. Factors are responsible for the transmission of characters, and the factors are present in pairs. So that's where you have to be very careful. Though these alleles or the factors are going to be present in pairs. Then law of dominance. In a pair of dissimilar alleles, if an individual consisting dissimilar alleles, not the same alleles, slightly altered alleles, then one will express and the other one will be suppressed. So the one that will be expressed is called as dominant allele, and the trait is said to be dominant trait. The one which could not express or its expression is suppressed, that is what is called as a recessive allele, and the trait is said to be recessive trait. Right. Then he also proposed the first law of segregation or law of purity of gametes. It states that when a pair of factors for a characters are brought together in a hybrid, they segregate during the formation of gametes. What does it mean? See, in hybrid, that means after the pure breeding plants are crossed, in F1 generation we got one plant which is having the heterozygous condition or which is having this dissimilar alleles. So they are together, but when gametogenesis will take place, the capital Y will move to the one gamete and the small y will move to the other. So that means they are going to segregate, separated during the formation of gametes. Even though they are together, they are going to get separated. So that is called as law of purity of gametes or law of segregation. Okay. Then dihybrid cross. Dihybrid cross, where Mendel just improved upon his experiment, he considered two traits at a time or two characters at a time. Say for example, the height of the plant and the color of the seeds. That means tall and dwarf are the two traits of our height of the plant, and the color of the seeds, round, and the not color, what we say, the shape of the seeds, that is round and the wrinkled seeds. Okay, together, how they are going to inherit. So this the study of inheritance of two characters which are responsible for this responsible by two genes, or study of inheritance of two genes will be called as dihybrid cross. Okay. So here Mendel selected pea plants having two pairs of characters. So here what he has selected is shape and color of the seed. Shape of the seed, that is two traits, round and wrinkled, and color of the seeds will be yellow and green. Okay. When he crossed these plants, what was the result he got? Okay. Here you can see. And while writing the genotype, so as usual he has taken the pure plants. That means there is no segregation for several generations. Such plants are called as pure breeding plants. He has selected as a parents, and for each trait how many alleles? We have to write two alleles. Suppose here in the parent he has taken round and yellow traits, right? So the plants having the round seed with yellow color. So how many alleles you have to write? For roundness, two alleles, capital R and capital R, and for yellowness, capital Y and capital Y. So they are together. So that means we have to consider them together. We don't write only one R and one Y, because each trait is controlled by two alleles. So capital RR, capital YY, that will represent the round and yellow seeds, and small r and small y. So that is, it represents the wrinkled seeds with the green color. Okay, so that's what. Then during gametogenesis also what will happen? From this pair, capital R and capital R, one R will move to the gametes. Okay, and among the capital Y and capital Y, one Y will move. So the gametes will have capital R and capital Y. Okay. So here the X or the other gametes will have small r and small y. So out of the pair, one will move to the gamete. Out of the pair of capital RR and pair of capital YY. Or in the same way here, small RR, small YY. In that case, so what will be the, so here the gametes will have capital R and capital Y, and here gametes will have small r and small y, and when they fuse they give capital R, capital Y and small y. Of course, when we write the alleles for a trait, we have to write together. Okay. So here in the F1 generation, we got all the plants with round and yellow seeds. This is the F1 generation, but having the different alleles from the parents. So what is that? In parents, capital RR and capital YY was there for round and yellow seeds. But here it is capital R and small r, and capital Y and small y are present. Okay. Then when the gametes are formed, you can work out the gametes. If you work out, there are four gametes, four possible types of gametes are there, right? So out of this, one parent, four possible gametes. So that means 25% of the gametes will have. Previously in case of monohybrid cross, what we are telling, 50% of the gametes, but here there will be 25%. 25% of the gametes will have capital R and capital Y, and 25% will have capital R and small y, and 25% will have small r capital Y, then the 25% will have small r small y, and the same type of gametes are produced from the other parent. This is for one parent, say the female gametes, and this is for male gametes. Same type of four types of gametes are formed. Okay. When we fertilize them, or when the fertilization takes place, all these gametes get all these alleles going to be mixed, and here we get capital RR, capital YY, capital RR, capital YY. So like that, there are this one is there, and if you see that there are 9 round and yellow colored seeds. 1, 2, 3, 4, 5, 6, 7, 8, 9. So the round and yellow will be 9. Then there are 3 round but green. 3 round and green. This is. And there are 3 wrinkled and yellow, wrinkled yellow. So here we find wrinkled yellow. 3 and 1 wrinkled green. So the phenotype ratio of F2 generation in dihybrid cross will be 9 is to 3 is to 3 is to 1 with respect to round yellow and then round green, wrinkled yellow and wrinkled green. So this is the phenotype ratio. This ratio you must be very careful. You have to remember both monohybrid phenotype ratio and the dihybrid phenotype ratio in F2 generation. Okay. So the same thing you can see here. It is the simplest form. Okay. Round green. This is the second what we say, round green, wrinkled green. We find 9 is to 3 is to 3 is to 1. So this is the example what we have discussed. Then how the expression of traits will take place? So generally the expression of trait will be taking place in this way. So DNA is there, a part of the DNA is a gene. So that particular DNA is going to be transcribed. So that process is transcription or is going to be transcribed into an mRNA. So this is DNA and this particular green colored structure is mRNA. So the information from DNA is copied to the mRNA by means of transcription, and from this a protein is going to be formed from the mRNA based on the information, based on the codes present on the mRNA, and that process is called as translation. So the genes are going to be expressed by transcription to mRNA and the translation to become protein. That is what is called as central dogma of the molecular theory, right? Molecular basis of the inheritance, central dogma. Okay, then we are going to discuss in the next chapter or next video, we are going to discuss about sex determination in case of some of the animals and in case of human beings. Okay, thank you. Wait for the next video.